maybe just I will adjust my main screen. Can I just share for the time being?
Yeah, you share, no problem, sir.
Maybe somebody has to enable me.
Yes, we will enable. I thought it is already enabled. Now it is enabled, you just see.
Okay. 1 second. Yeah, it is enabled. 1 second.
Good morning, Jagdish Garu. Dr. Jagdish, good morning. What is this PJTSA participation is not up to the mark?
Sir, already now World Pulses Day is going on, sir. Celebration, sir.
Oh, World Pulse Day today, eh?
Yes, sir. Yes, sir.
So that is the, I think. Good morning, Jagdish Garu.
Yes, sir. Good morning. Good morning.
Good morning, sir. Jagdish, sir.
Good morning. Good morning, sir.
Jagdish, good morning.
Good morning. Good morning.
What is this PJTSA participation is not up to the mark?
Sir, already now World Pulses Day is going on, sir. Celebrations.
Oh, World Pulse Day today, eh?
Yes, sir.
So that is the, I think.
Sir, today many programs are there, sir, at the same time.
Done, I think the 17th or 18th.
Sir, sir. Sir, basically, Raman, sir, I will come back.
Run. Very good morning, all of you. We'll wait for 5-10 minutes more. Done, I think the 17th or 18th. Yeah. I think somebody has seen.
Sir, sir.
Jagdish, చెప్పండి. వెళ్ళొచ్చేయండి సార్ ప్లీజ్. అదేంటి వెళ్ళట్లేదు, లైవ్ అవ్వట్లేదు. వెరీ గుడ్ మార్నింగ్ ఆల్ ఆఫ్ యు. వెయిట్ ఫర్ 5-10 మినిట్స్ మోర్. yes. నా మాట చాలా లేట్గా వస్తుందా? Yeah, Dr. Pradeep, Dr. Jyotsna, I think people are now joining. And Mr. Surendra also joining. Jakaullah Khan ji, good morning.
Good morning, sir.
How are you?
Fine, fine. How about you?
Yeah, I'm doing good.
Yeah.
Dr. Pooja Kumari, she joined back after the studies in IRA?
Yes.
Pooja, you are not.
Back on 2020.
Last year you joined, huh?
Yes, sir. I joined in February. Yes, sir.
But you have not told me. You thought that this man is no longer in NBPGR. You need not tell. You thought like that. Pooja? I'm still with you.
What, sir?
You thought that this gentleman, Dr. Babu, is no longer in NBPGR. So you did not want to tell me.
No, sir. It's not like that.
Like that. Good. So you are back again, doing your work, right?
Yes, sir. I have completed, sir, this PhD.
You have submitted your thesis?
Yes, sir. Degree awarded in 2020.
Awarded. Okay. You have
Yes,
taken
sir.
degree, last convocation.
Yes, sir. Last time.
Very good. Very nice. And there are about 20 people. Mariadas. Dr. Celia will wait for 5 more minutes, right?
Yes, yes.
घर रजिस्टर करके वीडियो दी से
Good morning, Suresh sir.
How are you, Pradeep?
Yeah, I'm doing good, sir. Thank you. How are you?
Good morning. Nice to see you after a long time.
Yes, sir. Same here.
Thank you for coming.
How are you?
Hello, Josna, how are you?
Great, sir. How are you?
Yeah, nice. I'm good. And it is nice to see you. And thanks for joining.
Yeah. Sure, sir. Of course. You are the one reason, and also the virus is the very reason to attending this call.
Okay.
Dr. Suresh, we are keeping you waiting.
I think we will
No
start.
problem, madam. No.
Suresh, is it all right for you?
Yeah, I'm ready. Yeah.
Yeah, you're right. We will just start in a minute. Encasing. Okay, good morning, all of you. This is Dr. Sharath Babu, President, Plant Protection Association of India. I'm very happy to welcome all the wonderful people today for this wonderful seminar from Dr. Suresh from CIMMYT. I think Dr. Celia is going to introduce the speaker, today's speaker, Dr. L.M. Suresh. In fact, we have started these, webinars, virtual talks in the backdrop of we have been in existence. Plant Protection Association of India has been in existence for the past 50 years. The society was initially started at NIPH, and those days it was CPPTI way back in 1972 in December. And now this year we are actually 50 years. So in this context, we have actually started this web series. This is actually today's talk is actually 7th, though it is being, in fact, we have had one talk in sometime in April 2021. And after that, on a regular basis, monthly, 1 or 2 talks from eminent people are being organized by the society. We have had in the past the first meeting, it was held on April 22nd. Is gene editing a myth or reality? CRISPR-Cas9 application in agriculture by Dr. Niranjan Kumar, President and CEO, ABS Incorporated, USA. And then the second one was on October 22nd, novel approaches for simplified detection of plant viruses and virus-like pathogens by Dr. Barnwal, IRA. The third one was space entomology, its significance in astronauts world by Dr. Ravi Kumar Asmani. Presently is an assistant professor in US Harvard. Formerly, he was a scientist at NASA. For a while, I think few years he was there, and then moved to on the invitation of Vice Chancellor. He has come back to India and is working with the US Harvard. In the series, we have had a talk from Dr. Vinod Kumar on plant protection in precision agriculture. With UAVs by Vinod Kumar, yes. Founder and CEO, Sensacre Labs on December 13th. And the fifth one was on plant biosecurity system for managing biological threat incursions by Dr. S.B. Sharma, the World Bioprotection Forum, Australia, on January 18th. And the sixth one, confluence of modern day pest and resistant pest management and resistant management by Dr. Srinivas Parimi, Asia Pacific lead, Bayer Crop Science, Singapore, on January 31st, 2022. And the present one, biosecuring Sub-Saharan Africa from Transboundary Maize lethal necrosis disease by Dr. Suresh from CIMMYT is this particular talk is arranged by Dr. Celia. I think Dr. Celia is going to. Also, I forgot to mention that we are actually organizing an international conference in November 2022. That is from 28th to 30th, 3 days program. Most likely, I think if the pandemic hopefully ends and we will have a physical, meetings. Else it is going to be a hybrid kind of, in the international conference. So I think it is the details are already on the website. And I think I request most of you, all of you, to participate in the international conference. Now I move on to Dr. Celia. Please welcome Dr. Suresh, and further the proceedings will go on. Thank you, Celia. Please go on.
So thank you, Dr. Shardababu. Very good morning, everyone. I welcome Dr. Suresh and all of you on behalf of Plant Protection Association of India and on my own behalf for today's lecture. And today's honorable speaker, Dr. Suresh, Dr. Shardababu, President, Plant Protection Association of India, Vice Presidents, Office Bearers of Plant Protection Association of India, senior colleagues and friends. And I have seen Dr. Dinesh Singh from Indian Phytopathological Society. Welcome, Dr. Dinesh, and all my colleagues and friends. It is my privilege to introduce today's speaker, Dr. L.M. Suresh, Maize Pathology Lead, Sub-Saharan Africa, CIMMYT, Nairobi, Kenya. Dr. Suresh has done BSc Agriculture and MSc Agriculture Microbiology in UAS Bangalore and PhD from Kevempu University, Shimoga, Karnataka. And surprisingly, he has done MBA also from IGNOU, New Delhi. And he began his career as Assistant Manager, Plant Protection, Indo-American Hybrid Seeds in 1995. And he was there up to 1997. Then he moved on to Nunhems India Private Limited as Plant Pathologist, 1997 to 2000. And then he joined CIMMYT Vegetable Seeds, 2001 to 2009. As Plant Pathology Lead, West Asia. And CIMMYT Vegetable Seeds, he has been promoted to Asia Lead Pathology Application. He was there up to 2011. Then again, he moved on to Monsanto Holdings Private Limited as Plant Health Application Lead from 2011 to 2014. And then he joined the CG Center, CIMMYT, as Maize Pathologist, Sub-Saharan Africa in 2015. And he is continuing there, and he has received many awards and honors, and 2021 Eminent Scientist of the Year by NESA, and NESA Scientist of the Year 2020, and Indian Phytopathological Best Research Paper Award in 2019, and he has received Appreciation Award in Achieving Research Excellence in Public and Private Partnership with Maize Necrosis Screening Facility from CIMMYT 2019. Can host the mute whoever is speaking? Yeah. And in Monsanto, he has been awarded several awards right from 2008 onwards. Every year I'm seeing his biodata. Achieve Excellence Award, Innovation Award, Appreciation Award, and many others. And Dr. Suresh currently is a maize pathologist, Sub-Saharan Africa and MLN Screening Facility Manager at CIMMYT, that is International Maize and Wheat Improvement Center in Nairobi, Kenya. Has earned professional recognition for breakthrough research in plant pathology and grading. A primary focus of his work has been the global Maize program projects that have strategic significance in maize pathology, disease diagnosis, epidemiology, and disease resistance, and uses of his scientific research findings as a tool to uplift the livelihoods of the 300 million Sub-Saharan African smallholder farmers that grow maize as a staple crop. Actually, this is the ultimate for any scientist who is working in pathology or entomology or as a breeder. If we can help the poor farmers, it gives a satisfaction to all of us. And his work has contributed to combat maize lethal necrosis. He has screened a huge number of germplasm, 220,000 germplasm from various public and private sectors. And kudos to you, Dr. Suresh. And he has his efforts have resulted in the release of 19 maize hybrids in Africa that are resistant to maize lethal necrosis disease in Sub-Saharan African continent. And he has also supported several of the national agricultural research and extension organizations in developing maize pathology laboratories, disease screening facilities for identifying and developing resistant hybrids. And he has also identified and validated disease trait donors with host plant resistance against many diseases, or MLN, MSV, maize chlorotic mottle virus, sugarcane mosaic virus, and also blight disease, and successfully developed and established comprehensive seed health program and policy for the entire CIMMYT maize germplasm in the entire Sub-Saharan African continent. And while working in private seed companies, he has developed and established pathology research programs with fungal, bacterial, viral research, with molecular ELISA and tissue culture labs, and associated in vegetable breeding. He has developed and optimized disease screening protocol for more than 22 diseases in 9 vegetable crops. And he has screened more than 50,000, all thousands only, not in hundreds, 50,000 germplasm received from all over the world in Aurangabad, Maharashtra. His support to breeding program has impacted in identifying and delivering disease resistance hybrids in many vegetable crops, very high value crops actually. Now I request Dr. Suresh to speak on biosecuring Sub-Saharan Africa from Transboundary maize lethal necrosis disease. Dr. Suresh, please.
Thank you, Dr. Celia, Madam. It's really, I'm honored and humbled to have the introduction. It is a great pleasure to be with you all, and also the President of Plant Protection of Association of India, and also the member secretaries, vice president, and all the office bearers. I'm immensely thankful to all of you for giving a great opportunity to share the work what CIMMYT has been doing. Today, I will be presenting on behalf of all the colleagues. The work has been done in interaction with the partners. So together as a teamwork, we could be able to do some good work to support the livelihood of farmers in Africa. So I'll just start the presentation. So without taking much time, I hope you all can see the slides. Can you see the slides? Yeah, can you see my slides?
That's right.
Yes.
Yes, we can see.
Yes, we can see the slides.
Okay, okay. So again, okay. So today I'll be presenting the topic on biosecuring Sub-Saharan Africa from transboundary maize. Just a second. Yeah. Okay. Transboundary maize lethal necrosis. especially the work we have been carrying out in Africa. So this virus is new to the African country last several few years. But before that, I would like to give you a brief on the maize. Maize is Africa almost to the tune of 35 million hectares, and they're producing almost 70 million tons. So an average of less than 5 maize is consumed. Almost 85% of the maize is consumed as a food by the farmers and also other people in Africa. That means you can see the livelihood is mainly on maize. The low and then the less activity is mainly attributed to several biotic and abiotic constraints, and mainly the biotic constraints due to the diseases, various diseases such as maize lethal necrosis, maize streak virus, other foliar diseases, and ear rot, several diseases. And fall armyworm is one of the major constraints also as an insect pest. So besides drought, low nitrogen use efficiency, those programs we have been handling. So with all this, they are finding a very challenge to grow maize, but in spite of it, they are depending on the maize. As I mentioned, the maize is been affected by various diseases, such as Triticum leaf blight is a foliar disease, gray leaf spot, Fusarium ear rot. Of course, ear rot is a complex disease. So one of the major causal organism is Fusarium ear rot, and common rust virus is the main disease in entire Sub-Saharan Africa, especially in East Africa. Maize lethal necrosis is the main disease. So to combat this biotic stress, the major approach is to have a resistance to all these diseases. Then only farmers can produce the maize and then have the food. As I mentioned earlier, just a second. Yeah. So as I mentioned, this particular maize disease is a major complex disease affecting the maize crop, and this particular maize disease is tackled with the various institutions with multidiscipline to handle this disease. So main focus which I'll be covering today, that how we could handle this approach by intensive screening of germplasm and fast tracking the development and deployment of resistant tolerant varieties in the entire Africa adopted genetic backgrounds. The next most important.
Sir, maybe you can make it duplicate
Excuse me?
slide. Sir, you can make it duplicate slide. We are seeing two slides.
Yeah. Okay. Is it okay now?
Yes. Yeah.
Yeah. This is good. And the second, thank you, Josna. And second is that optimizing the diagnostic protocol for MLN causing viruses, especially MCMV and SCMV, and also MLN. So the most important combination of viral inoculum, MCMV and SCMV that I'll be covering. And we are coordinating with the public-private partnership. Most of the, even a recent visit to my India, they feel like a public-private partnership is not a success. But here is one of the key examples to highlight how this can be handled with public-private partnership. And also we are focusing on MLN diagnostic and disease management. Next is MLN monitoring and surveillance across entire Sub-Saharan Africa in collaboration with national plant protection organizations. And now also the under one CGR umbrella, plant health initiative is focusing on again on this disease. And this area of surveillance and diagnosis. And mainly we are focusing on partnership with private partnership on the production and exchange of MLN-free germplasms and most MLN management, including the policy makers. That is the key part. So as you all know that, I think most of you are aware that maize lethal necrosis is a viral disease, and it is not, MLN is not a pathogen. It is a disease, and it is affected by maize in combination with the maize chlorotic mottle virus and any of the party virus. In this particular case, sugarcane mosaic is a virus is the common one. Sugarcane mosaic virus, for your information, is existing in Africa for the last 2-3 decades. So it was not affecting. But when the combination of maize chlorotic mottle virus, it is causing a devastating effect. The disease was reported in Africa, particularly in Kenya, during September 11. Since then, the disease was reported in Uganda, Tanzania, Rwanda, DR Congo, and Ethiopia. So, as you are aware that most of our food crops being affected mainly several diseases and pests. So, in this case, especially in maize, so many pests and diseases we can see here. So these diseases and pests are contributing to the crop loss of almost 22% in the global. But in Africa, the major pest and diseases affecting the maize is maize lethal necrosis and fall armyworm, other than the other diseases of wheat rust and fusarium wilt in banana. So this particular disease was understood in terms of economical damage by Dr. Hugo et al., our colleague in CIMMYT, and he did a study during 2016. Initially, the crop loss was quite high, and especially he made a study in Kenya. It was almost affecting to the tune of $188 million worth alone in Kenya. Imagine other countries like Ethiopia, Tanzania, Uganda, Rwanda, and DR Congo, Burundi, and then they have been affecting so much. But later, in 2021, again, the same team has studied the disease has been reduced quite significantly due to various interventions. And you all are aware that, as I mentioned earlier, maize lethal necrosis is a disease which is a combination of two viral diseases, maize chlorotic mottle virus, any of the virus. In this case, sugarcane mosaic virus is the common. Alone, they also can make a disease, but in combination, they are very lethal to the maize, especially at early stage. So the typical affecting the crop is seen as a mosaic, but later it is completely showing death of the plant, but depending on the germplasm resistance mechanism. So in this particular case, the typical symptoms is starting with chlorotic stripes, and then slowly starting with chlorosis and marginal necrosis, stunting of the internodal length, and the poor grain setting, the kernel you can see. But sometimes we can also see rotting of the cob due to other factors. The disease is not new just in Africa. It was reported in Peru 1973, later in USA, Argentina, Brazil, Thailand, and Mexico, Hawaii, China. And then it has reported in Africa 2011. And 2011 to 2016, it has been in Africa, and later it is also observed in Ecuador and Spain. And thank God it was not reported anywhere. Otherwise, it is. You may be wondering why this particular maize lethal necrosis is very damaging in Eastern Africa. Especially, as I mentioned earlier, sugarcane mosaic virus was there, but MCMV was new to the region. Potentially, the combination of new strains of SCMV. You may be aware that the agroecology in Africa is very conducive for any crop in growing. Especially, they take up the maize almost throughout the year. You can, so that region to get a continuous build-up. And seed contaminations. I'll touch base on the contamination and transmission later. Especially the MCME virus is mainly contaminating and making it the local spread through insect vectors. And most importantly, they don't have the resistance germplasm that has been widely cultivated in the entire agroecologies. So with all these reasons, the large proportion of commercial maize varieties are very susceptible making a crop is very vulnerable to MLN. So we did not just follow only one practice, deploying, developing and deploying the resistance. We had several strategies to tackle this viral disease, especially understanding the epidemiology with innovation and also engaged several multiple stakeholders in Africa, especially AATF Agra for MLN-free seed monitoring and surveillance, especially with the help of NPPOs. And we are also training several partners to have good agronomic management practices. One of the key areas is that to have a community of practices. With all this, we are able to at least see the disease under check in this Eastern Africa, and also preventing further spread from Eastern Africa to Southern Africa. So you can see in 2011, a farmer was really having no hope and getting no yield from their field. The similar farmers, they are very happy getting the good resistant hybrid, and now they are able to harvest at least 6 to 8 tons in a very successful cultivation, but at least they have a better yield in the heart disease pressure. So as Dr. Cecilia Madam was mentioning that we have been screening the germplasm from 2013, but 2014 we moved to centralized screening facilities. So we have been screening more than 215,000 germplasms coming from all over Africa, including also we are receiving from India sometimes, and then Mexico and other countries. Large proportion of this germplasm is coming from.
Can you switch off the video so your internet maybe would be better?
I think that's a good suggestion. That's a good suggestion, madam.
Yeah. Okay, thank you.
Yeah, and so in this case, a large proportion of the germplasm is coming from CIMMYT and also coming from NARS, especially in 8 African countries, and small and medium enterprise companies around 20%. It is not just screening the germplasm. To maintain the high quality of data and turnover with the qualities is the key. We are getting the data quality almost 0.99 heritabilities consistently, including some of the molecular studies data, gene editing the trials. All the data is very consistent. Continuous improvement. We are not just there to screen, but we also supported the various platforms, including MABC programs, molecular acid, bacterial acid programs, for converting most susceptible conversion for more than 50 elite lines. So as I was mentioning to you, you can see this is the screening facility which we have in Kenya. And all the 4 sites, you can see the entire facility was like this kind of bushes before, but we developed this entire facility like a very systematic quarantine facility, and it is a quite stringent quarantine facilities. So I will not take much time, but to give you a few idea about the features of these facilities. We have a lab facility and pure culture maintenance facilities and incineration. Whatever the plant material or anything which is used here will be completely incinerated here. Nothing will be going out. So that is another thing. And media filling and sterilization facilities and environmental control greenhouse facilities, and especially here automated fertigation drip irrigation facilities. The entire area is completely supported by automated fertigation drip irrigation, so that we which this facility is 20 hectares, out of which 3 hectares is dedicated to facility and 17 hectares dedicated to farming. And please understand that people do claim that there are several germplasm, they are resistant in America, but when they are screened here, we found 100% susceptible here. That means the environment is quite conducive, the pathogen is quite. These varieties could hold well in other countries like Tanzania and Ethiopia, so that this kind of the rigorous screening is supporting the sustainability of the product deployment. So it is not just facility. We have a very thorough screening facilities in terms of consistent discipline protocol. As I mentioned, these are all the facilities, and process is also well established. You can see here our system of handling the germplasm from partners. And then second is the automated fertigation, drip irrigation facilities, and use of handheld for data recording. Even we were working on the drone support to take the data. And also well laid out phytosanitary procedures for different viruses and also incinerations. So we do screening for almost 5 times a year to support the partners. It is not just to say for 5 times. Why 5 times means it is to support the breeding program fast tracking mode. And second, to support many people in the world to send their germplasm any time of the year, so that we don't know at what part of the year they are producing seeds, and they can be easily access to this facility any time of the year and go back to the research program and fast track their product development. And we developed a very thorough screening protocol with 1 to 9 scales, 1 being completely clean and 9 being completely dead. This is disease screening protocol is mainly for lines, and this is mainly for hybrids. So far, we find many entries on the scale of 2, and when I joined at that time, most of the germplasm 6 or 7. You can imagine 6 and 7 what it looks like, and 80% of the germplasm looks like this. Now at Touchwood, the most of the germplasm is holding this standard, clearly visible. If you visit today or tomorrow, it can be easily observed. That is the beauty of our breeding program. This particular slide is very important. Most of the African breeding partners, extensively they are using this particular few lines. Invariably, they are all drought tolerant and tolerant to various other diseases. And now they are highly susceptible for MLN. But with the continuous improvement, we have converted them into resistant. I will explain in a minute. So our key source of resistance is a case 23-6, which is coming from Kisat State University in Thailand. So that is how we have been successful in developing and deploying the resistant hybrids. So over years, we could make a lot of progress. As I was telling, when I joined at that time, the mean was almost 6.7, 6.5 for the larger germplasms. And it was almost 5 in 2018. I can say confidently now the mean has shifted to almost 3. This particular study was carried out by Dr. Bayane Yosef, and he's our breeder in Eastern Africa. He's focusing on this particular product profile. So you may wonder, this particular resistance is polygenic recessive, having additive dominant effect, and also some of them are complementary in nature. So we don't just screen in facility, as I mentioned, we also take Africa. In Ethiopia, especially in Babati is one of the hotspot. Even then, our hybrids are quite good in under the high disease pressure. So when we screen in Wandagonet in Ethiopia, the disease pressure is very high. Under that circumstance, also our hybrids are doing quite well. That means you can see it is not just to say that we screen and deploy, but we also validate them in a various hotspots. Then we subject for product deployment phase. So when we study in then optimal conditions, the yield is fantastic for those hybrids. So this is how we can see the performance. As you can see here, the commercial hybrid having almost 77% crop loss, whereas our second generation having 3.1% crop loss. So we are still coming better the hybrids with better hybrids, but they are very good. And with this kind of efforts, we have identified almost 19 hybrids, and we have already released to the Eastern African Community partners. Now they're marketing. And one of the key examples is Bazooka, which is drought tolerant and also MLN tolerant. And earlier, you can see most of our commercial hybrids are like this. There is absolutely no yield. And you can imagine, even now also they are being available in the market. But against to this, we started with the first generation tolerant, whatever the germplasm available, including some of the commercial checks. And now we have identified several other hybrids which are very good in terms of yield and the kernel qualities, adaptabilities, and maturity, and various other agronomic characters. So some of the companies in Kenya, they have produced more than 500 acres of seed, and now getting it good success for the seed companies. And second is that they need not to invest a lot of money on the research, where CIMMYT is in their backdrop. They can easily take the support of CIMMYT. And I will switch over to another slide on the molecular breeding. So as I was mentioning to you all, that this particular resistance program is started with the donor case 23-6. This particular line having very good level of resistance. Even now it is supporting for a program. So especially this line, when molecular work has been done by Dr. Manjagowda, and he has done a very good work on understanding and also on the genetic architecture of the MCMV, and he identified chromosome C, the QTL for MLN, so which has been quite used in various programs. One of the program is molecular assisted backlash program. A few seconds before or a few minutes before, I was telling you, this is the line which is widely used across all the Africa pipelines in various companies. And it is quite good, but it is highly susceptible to MLN. Today, we converted the same line and making it MLN resistant. This is one of the key strengths of the breeding program, and in association with the marker and pathology program. This is how the continuous teamwork for the same cause. And similarly, several other lines like this, we have identified more than 52 elite lines. Now they are into resistant pipeline. So this is not just breeding alone. It is very important to understand how we curb the disease from the endemic to non-endemic. This is a facility established in Harare recently, almost 2016. And in this facility, when we send the seeds from Kenya, and that seed is completely free from MLN or MCMV, the germplasm is grown in this facility, and it will be monitored for entire one season. Imagine this field, if there is one plant is identified, the government of Zimbabwe, they are going to destroy the crop around 50 kilometers from entire this area. So they have taken stringent measures, and this facility is within inside the National Plant Protection Organization's campus. So you must understand that the difference between quarantine and screening facility. In the quarantine facility, we do not inoculate, but we still monitor and conduct the survey. Whereas in screening facility, we do inoculate, we do screen and germplasms. But in this case, we do not self or cross the hybrids, and then we just focus on the screening, getting the data only. And this is also quarantine facility and screening facility, but this is only quarantine facilities. So this is the two distinct feature, but CIMMYT is managing both the facility with strict quarantine measures. As I was telling you before, as Dr. Celia Madam was mentioning, that how we could support the seed exchange and distribution policies in entire Sub-Saharan Africa. What we do first, we scout in the field in our Kenya research farm, and we look for any symptomatic plants and diagnose them, and also we monitor. And before shipping the seeds, all the entries will be diagnosed at our lab in Kenya. And after we find that the entire lot is free, then we give it for Kenya Plant Health Inspectorate Services for testing. Once they find it free, then we ship it to Harare. And there they multiply. Again, they test, and then they distribute. And all our germplasm is following this flow of movement. Especially, it is started from 2016, and now we have tested more than 40,000 entries, and touch wood, there are no single entry found positive wherever the seed has been received from Kenya. And that is one of the key feature of this seed health lab. You can see here, the Eastern Africa still having the MLN, and Southern Africa is completely free from MLN. One of the key steps, this step is helping to prevent the disease spread. So one of the channel we need to look at it, how we can control the contaminated seeds from endemic to non-endemic area. In this particular slide, I would like to emphasize that the virus is not inside the seed here. It is not in the embryo or scutellum, endosperm, anywhere. But you can see here the gluma, the virus is just here. So if we disinfect in this portion, the seeds will be completely clean. Also, we consider as it is a contamination. It is not like a seed-borne diseases. So this particular understanding is very important. Seed contamination and seed-borne diseases. Not all seed contaminated is seed-borne. Yeah. So we should have this clarity before we make larger communications. And through engagement with the various other projects, we also focusing on the disease diagnosis and surveillance in the entire Sub-Saharan Africa. And especially when we do such disease diagnosis and surveillance, we continuously sensitize and support National Plant Protection Organization, National Research Organization in the entire 8 countries of Sub-Saharan Africa. So next part is that how we could influence commercial seed sector in MLN endemic countries for producing MLN commercial seeds to promote the use of clean hybrid seeds. That is the most important step. And also we developed a community of practices in the entire Eastern Africa countries. So in this project, I would like to emphasize here that we use simple step to diagnose the disease. We used immunostrips. In the field, it is most efficient, effective way to diagnose the diseases, because we just want to know whether the virus is there or not. Once the virus is identified in non-endemic countries, immediately we mobilize the samples to a very reputed lab to confirm it either by ELISA or RT-PCR. But in the field level, we just use immunostrips. All this information, what we train the people using ODK software in the mobile phone, they constantly provide all the details, including the image of the result, as well as the picture of the plants. So all this information is centrally available in MLN web portal, which is publicly observed. We do conduct such diagnosis and surveillance in various Eastern Africa countries and Southern Africa countries. These are the few examples where I am sharing with you. Just to give an example, this entire diagnostic step is being done within almost 15 to 20 minutes or half an hour maximum. So from the point of collection of samples and making them prepared and diagnosed, and once the sample is identified with virus, we can find 2 strips. If there is no virus, only 1 band. And if it is error, there is no band at all. So all this protocol is available in our technical manual. We have recently prepared, and now it's available in the public domain. So this kind of diagnosis we used in various kind of production. So this kind of surveillance program is quite helpful. Then we can take the necessary steps to control or manage the disease. So in 2014, the disease just more focusing on Kenya, and 20 and Tanzania, and also part of Uganda. The disease was furthermore was in Burundi, and then some of the Kenya, and then 17 it is again still there, and 18 it is still there. It has not been controlled. But all these years, because there are two reasons. One of the reason is that the effective intervention from various stakeholders, and also safe germplasm movement. And then you can see in Tanzania border, this particular zone is not much grown with the maize. This kind of vacuum is also preventing the movement of viral diseases from this portion to here. So this kind of natural physical barrier is also helpful sometimes to check the diseases. And with this kind of interventions, whatever the effort, whatever we are carrying out, all this information, protocols, steps, guidelines, we have compiled in a technical manual, which you can see in our web portal. And then while doing this kind of surveillance programs, we trained all the NPPOs. You know, they can go to any corner of the field, diagnose, and move on. And this is not very expensive, very simple and efficient, and fast in terms of updating the data. Within 1 week or 2 weeks, all these data will be available in central web portal. So this kind of information is available. And second is that we are not just using on the commercial seed farms or commercial farms, but also we are checking the seeds coming from various countries with agri dealers. So all the NPPOs will visit agri dealers, take the samples, check the disease, and this kind of information is available in the web portal. So it is not enough. So we have to help all the commercial seed companies to produce disease-free seeds, especially in Eastern Africa. Just to quote an example, last year, one of the seed company in Rwanda, they were having a challenge. They produced the seeds to the tune of 500 metric tons. And the government is saying that there could be suspected seed lot. The company was so worried how to handle that much seed. Otherwise, they have to simply burn it. But thank God, with all our support, and they could identify and test and come out with clean, because they have followed all the procedures, whatever is required for producing disease-free seeds. And to accomplish this kind of objective, we don't just say to them, we intensively conduct training at various points in Sub-Saharan Africa and make sure that they follow adopted practices. And this is kind of practices we developed, and it is supported by AATF in Africa. They do intensive training and monitoring. This kind of protocol is established. And if you see, these are all various steps. This is very simple principles of pathologies, but to make it to happen is very important and very difficult sometimes. So we follow these steps very carefully. Avoidance, especially to prevent the movement of people, tools from the infected field to a healthy field. And we advocate not to feed the infected plants to the livestock, and also have this discussed thoroughly with community relevant measures and exclusion of inoculum from in the field, either tools or contaminated seeds, and then eradication, remove the infected plants, and also use of pesticides. And also, most importantly, we rigorously support them to have a host-free period and support on crop rotations. In case if there are suspected plants, you immediately burn, and also grow the crop with a barrier of non-maize crop. and use of resistant varieties and disinfectant tools and implements. These are all very necessary, but looks very simple, without which we can't manage the disease. I would definitely believe and advocate. So all this information, what we have been discussing, they're all available in various languages, English, Swahili, Amharic, Spanish, so that every farmers can understand and follow this step carefully. So this is just some of the numbers, how we are supporting. And especially for helping the farmers in terms of producing disease-free seeds, we are being supporting to educate them how to follow the steps so that they can remove the contaminated seeds in the entire endemic areas. And several seed companies are now following it with all these SOPs, and we have been training all the partners. So these are some of the examples how we have strengthened the capacity of the partners, and now they are completely independent and taking up the task to the standard. And also this year onwards, again, we are making more efforts with the one CGAR plant health initiative under the leadership of Dr. Prasanna. And now we are able to reach out again, help them so that the disease can be managed. For your information, disease is still there. We need to handle it to the last point. And these are all the various examples of how we conduct the training in different countries. So most importantly is that we need to sensitize the stakeholders and also policy makers. So one of the key area is that community of practice. I think in every area or every country, every farms, they can make this kind of practice. Once in a while, they come and discuss on the disease and how they are following the disease diagnostic protocol, adoption of various steps. And it helps in terms of seeking an agreement on the phytosanitary committee of requirements, and also support or seek a cooperation on various activities that don't cooperate, and share the learning across the borders. It's not within the countries. And as you know that many transboundary areas, especially porous transboundary countries, they do exchange a lot of grains, which is very difficult to control, especially to sensitize. Having a strong policy and the borders which to check the disease is very important. So we try to help them to have a good policies, but still there is a lot of work to be done. And identify the linkage and opportunities is another area, and report on progress, whatever they have getting on the timely manner. And they also provide the information on the different SOPs. With this kind of areas, we continuously support in terms of policy making, and this kind of information is available for various government for having their uniform policies across the region. And all this information, what we are been discussing, is constantly updated and available to the larger public to know how things are done, and also various publications, manuals, procedures, reports, everything is available. You are free to browse. In case any questions, you can always come back. It is not just enough. We are also supporting and gene editing programs, especially on the fine mapping and cloning editing. So Carteva, with the help of Carteva company, so we are able to have this event, and the product is almost ready. Now we are going to have it in our screening facility for final validations. So once that is available, that will be further going to the larger public. So that is another latest update. So what is the highlight from all this information what I have been telling? So maize lethal necrosis is emerged as a serious threat to maize productions, and especially for the livelihood of smallholders. An intensive multidisciplinary and multi-institutional strategies being adopted, implemented to curb the spread of maize lethal necrosis in Sub-Saharan Africa, to mitigate the impact of diseases. So we have been taking up an intensive program on screening germplasms, and also a constant effort is being made with the multi-crested breeding. And then we have developed 52 L8 lines. And also a constant effort has been taken up for the diagnostic protocol, and which has been optimized. and this information is available
Dr. Suresh, I think there is a big problem with the internet from your side. But anyhow, continue. We are not able to hear you. I request all the listeners to kindly bear with, Dr. Suresh. Let us bear with him and wait for a few more seconds for him to come back again.
I think that must be his last slide, fortunately.
No, but that's good account already done. But I thought there must be some interaction. No, there are many people I think with questions. So let me try if you can get him on the WhatsApp.
Okay.
He is back.
Am I audible?
And Dr. Suresh, I think there is a problem with your internet. You are back. Are you able to hear me?
Hello.
Suresh.
Yeah, sorry for technical challenges.
I think internet there is a problem there, right?
Yeah, sometimes this.
I think Dr. Suresh, if you can conclude it, I think there are many people who are willing to ask you questions. I think.
No, I'm in the last slide, which you can see.
Please go on. We are only worried about your internet. That's it. We are not holding you.
No.
Please go on.
No. So I can say that this entire information is being published, available in the public domain, and perhaps I will not take much more time. I will request the audience to have interactions. Let's keep it more informal, more interactive. I sincerely thank the organizer for giving me this immense opportunity to share our experience. And in case if some disease comes to that kind of area, how can we address it together as a team?
You're done, Suresh?
Yeah.
Oh, excellent. Congratulations, Dr. Suresh. It is really a wonderful account of your work on MLN. It is really, I think many people do not know much about this disease because I think it is a quarantine significant disease in our country, in India. I think I was just talking to Mr. Sunil. He also told me that there are hardly any reports or no reports. So I think to begin with, I would like to know a little bit about the kind of germplasm exchange that is happening with Africa and India. What kind of I don't have information. My colleagues should be able to tell me what is happening with the exchange, and if at all it is happening, what is the risk of the MLN entering into India, and whether there are any encounters with this disease in the past in India, and what is that India has to look forward, India has to do. I also know that a lot of exchange is happening with the industry from African continent. So I'm really worried what is happening, whether the disease is reported or not reported or not being, really studied. I don't know, but I think you can give your idea on this. Let us have some more scientists from India also. I think they want to ask you also. Please come in.
Hope I'm now audible as you can hear from me. So thank you, Doctor. It is very important questions. I do have the same concern as you are. as I'm coming from the major maize growing areas. So the most important step that when we send the germplasm to India, we are not sending from Kenya as an endemic countries. We are sending, having it quarantined at Harare in Zimbabwe. So having it properly scouted, having it tested, once it is found free, then only we ship the germplasm from Zimbabwe to India. And so far, whoever has requested the germplasm from India, we are not at all giving any single seed from Kenya office to India anytime, because I'm the one who is constantly checking and monitoring the movement of seeds. Several requests have come, but we do honor the request, but following the guidelines. Number one. Number two is that so far I understand Indian quarantine systems are also quite vigilant in understanding the movement of seeds. Always Dr. Celia Madam asked, Suresh, what shall we do in case if I found the suspect at large? So we immediately advise, in case if there is found positive, please incinerate it. There is no mercy for it. Not a single germplasm at any given cost. If it is found infected, we do the same practice, just incinerate. And God forbid, it should not come to India. In any case, if something comes to India, as you all know, this is a pandemic era, and we have a kind of emergency preparedness, and also one should have a quarantine facilities, strict quarantine facilities, and then diagnosis and then surveillance mechanisms. All these steps, whatever we have been making here in Kenya, which can be adopted in India. So with that confidence, I think Indian scientists are quite brilliant and can do much better, and then they can tackle easily with all the multiple stakeholders. Especially, I believe that having worked in private companies, they do follow all the guidelines. I think teamwork can only a solution for this disease.
Sir, may I
Yes, NBPGR certainly is very capable of handling this.
Sir, may I submit, sir?
Please,
so
please,
for excellent
please.
presentation, Suresh, sir. We
Please
are always touch with CIMMYT Hyderabad team, Dr. Vivek, Jaidev, and then Dr. Pasan also. So most of the things, wherever we indent CIMMYT germplasm, we are not getting from Nairobi or Zimbabwe also. So this is the one thing. Also, mostly we'll get from the CIMMYT headquarters only, the germplasm. So that is the one thing we are doing, sir. And most of the things, wherever the germplasm comes, NBPGR Hyderabad is going for a post-quarantine evaluation. So we are taking care of all these things because Prasanna mentions once FA came, again MLN is the one thing, and then another is the tar spot complex. This is the two things we
those are the emergency.
We must be very careful regarding the post-quarantine evaluation. That is nicely taken care of by NBPGR Hyderabad, sir. Thank you very much.
Okay, yeah.
Very good point, Shekhar. I think Dr. Celia would like to come in. I think NBPGR, of course, certainly we are only concerned about the, post-entry quarantine, facilities of the industry, actually. I think at this point in time, NBPGR has to be more vigilant in, making regular inspections of post-entry in the, industrial, the post-entry facilities of the industry. So I think Dr. Celia, please come in.
Yeah, all the maize germplasm which come into the country, whether it is Hyderabad or Delhi, we do the post-entry quarantine inspections. And its germplasm is imported by both public and private sector, but private sector imports most of the germplasm, and I have appreciation for all the MNCs also, like they follow all the standard protocol, and if we are asking them to destroy the material, they are destroying it. And we don't have the MCMV, maize chlorotic mottle virus in India, and it is a quarantine virus for us. And we have intercepted this virus in germplasm imported by one of the private companies from Puerto Rico, Thailand, and USA, not from CIMMYT material. So in one of the, that is way back in 2010, actually, I was testing the healthy samples so as to keep it as a negative control, but we got the maize chlorotic mottle virus, and the entire consignment was harvested and incinerated in my presence. So absolutely all the MNCs and the local private companies, they are following the protocols, and both Delhi and Hyderabad scientists undertake the post-entry quarantine inspections, whether it is Aurangabad or Gujarat or Hyderabad, Bangalore, wherever it is. No germplasm is released without PUQ inspections, and whenever there are symptomatic plants, even before testing, we uproot and destroy the plants.
Thank you, madam. Only one which I can mention, some of the countries in Kenya and Uganda, and some of them, they are expressing many times they do not see symptoms in the growing field, but still they observe MCMV. Okay. So observation is quite valid that most of the times we do have to test, just not rely on the symptoms, to be sure that this is not just escaping any manner.
Yeah, we do test.
It's a Dr. Celia, can we just have those kits that these people are having at CIMMYT, so that we can also do that kind of testing at NBPGR? Because if Dr. Suresh says that it may not be expressing sometimes in the post-entry facility.
Yeah, those strips are available. Dr. Suresh, I think for me it looks like they are from Agdia, right?
Again, Agdia and Bioriba both.
Yeah, they are available. They are not CIMMYT strips. They are manufactured by the Agdia.
Publicly available.
Publicly available.
I think they should be used by NBPG, Dr. Celia. I think whenever Maize is there, I think Maize comes in.
We use ELISA and PCR. Like strips are.
So ELISA takes care.
But it becomes a little expensive. The strips are.
Okay.
Yeah, we do testing at Delhi.
Oh, but if it is industries, I think we can charge them also. That may not be a problem. But I think we must take care of this disease.
Yeah, we are taking care, and we can discuss later. Let's know. Yeah.
Okay. Dr. Celia, because Dr. Jagadishwar is not there, why don't you ask, most of the senior persons are there to come in and, give his remarks. Because Dr.
If you can raise your electronic hand, I can take the questions, or you put in the chat box.
Yeah.
And Dr. Ahuja is writing, this is an interesting story and scientific efforts to disseminate the proven technology for distribution and production of MLN disease-free seed in the Africa. Congratulations. And Dr. Muhammad is asking, I would like to ask you that whether you tried any chemically induced resistance of the crop. Please describe somewhat. It is there in the chat.
Yeah, of course. I will take up this question. Thank you, Dr. Muhammad. So far, native genetic resistance is the most focus we have been doing, and followed by the gene-edited project. That is another point. So I think these two steps is helping us a lot with the simpler steps. And then also a lot of effort has been done by molecular breeding team to convert susceptible into resistant. So they're holding a high level of resistance. So that will be the good examples to support the breeding program.
Okay, thank you, Dr. Suresh. Dr. Pradeep Kumar wants to ask the question. Dr. Pradeep, please.
Yeah, thank you. Suresh sir, it was a nice presentation and very informative. Thank you for that. And since MLN is a seed transmitted, in most of the vegetable crops, we find many seed transmitted diseases. And for some of the diseases, seed disinfection will work. Similarly, for MLN, any kind of research where we can use seed disinfection. Why I'm asking is like there will be a lot of effort in growing crop and producing seed. And then directly, if there is a MLN detected in the seed lot, destroying all seed lot, yes, that is one option, but we'll be losing a lot of time, effort, money, so many things. So can we use any disinfection kind of thing, any kind of research CIMMYT is doing on that?
Okay, thank you, Pradeep. This is one of the important questions always I used to discuss with Dr. Celia Madam. But here, that this is a seed contaminated, it's not a seed transmitted. Yeah. And this can be easily disinfected using some of the chemical, say, for example, trisodium orthophosphate. The challenge is not just treating the chemicals. What happens, we have taken up a study using the 500 to 800 metric tons of seeds and different seed lots. The challenge is not that. challenge is that any point of time, if the contaminated seeds and treated seeds are getting mixed up anywhere in the kernel, one seed is enough. Imagine they say 0.03% or sometimes 6 plants in a hectare is enough to again bring back the diseases. We want like 100% check. It is not easy to get 100%. Although I have tried TSP for almost consecutive 3 to 4 years, I can confidently say it will be helpful. But any point of time, there will be a kind of contamination during the handling process. So which I don't want to advocate getting into a challenge and bringing back the disease in the non-endemic area.
Yeah, thank you, Dr. Suresh. I think Dr. Pradeep is asking something like 10% trisodium orthophosphate we usually do for tomato, chili, tobacco. So
Based on that only
comes for MLN, it would be really great because all the germplasm coming from AVRDC Taiwan is disinfected with 10% trisodium and come into the country, or if they don't disinfect, we do it at Delhi before we release. That of course is
that is a good
only for Tabama viruses, not for other viruses.
Yeah, this can be used, madam, but only my caution is that sometimes and large seed, especially vegetable seed, is in small volumes.
Yes.
That is where only the caution we finally realized that will be a very great risk. For germplasm handling, small quantities is absolutely fine.
So do you recommend 10% trisodium orthophosphate treatment for germplasm if it is 20-30 seeds?
Yeah, it is advisable, but we did not make as a kind of a regular policy measures so that we don't want to get into a risk again. But it can be used, think of to adopt.
Okay, thank you. Thank you, Dr. Suresh. Dr. Sunil, please.
Sir, a very nice presentation and very relevant since in the context of the recently we were challenged by the FAW infestation in 2018. So we need to be prepared for such a disease which may enter into the country. But I'm particularly more interested in this resistance source that you have mentioned, sir, whether it is being shared with India, whether it is shared with our people, or whether it has been available for our use, crop improvement in maize, KS 23-6.
Yeah, it is available. It is generally we got it from the Thailand University, Keset Start University. I'm sure that they are also giving it to any other people. And also the advanced germplasm with the better level of resistance, good the kernel qualities, those maize are also available from CIMMYT with resistance. So there is another step, Indian breeding program, especially Vivek, Zaidi, and Sudha, they are also working on preemptive breeding strategies, meaning that they want to have a screening in Kenya and having it integrated with the recent lines. So once that has been integrated, because the India is using the yellow germplasms and Africa is using a white germplasms, and these are the two distinct phases. So this can be done easily, so it can be adopted and deployed.
Thank you, sir.
Thank you, Dr. Suresh. And Syngenta imports lots of maize germplasm. I see Dr. Lovelin. If you are from Syngenta, if you want to ask anything, please ask. Anyway, let her come back. Professor K. Jeevan Rao is here. If you want to ask anything, sir. No. Okay. If there are no questions, Dr. Suresh, I have a small queries. Like you mentioned, collaboration with NPPOs. How it happens? Because actually in India, NPPO collaborating with us, they only ask us to test the samples. So all this, how it is done there in Sub-Saharan Africa?
Especially in Sub-Saharan Africa, we do collaborate with NPPOs, especially in terms of disease diagnosis and surveillance, and also to give a technical backstopping to have them very good protocols for checking the commercial seed productions. So earlier in Kenya, especially, they used to have a standard of 10% acceptable limit. So when they have been constantly educating, they come to the point of 1% tolerance limit. Again, we sensitized and realized the situation and make them aware that this is how we have to do it. Now it's 0% tolerance. Imagine a plant in an entire 1 hectare land, if they found it is completely rejected, no matter what company has anything. So that kind of a constant engagement is happening. And also, in case if there are any issues with quarantine norms, we do provide technical backstopping to them, not just in Kenya, Ethiopia, Uganda, Tanzania, all the Eastern and Southern African countries. So that is how constantly we support. Whenever such grant comes, we do support them in terms of diagnostic kits and also facilitate them having this surveillance is done. So we do acknowledgement and credit for the good work they have been doing. So this is how we have been supporting them, and they have been requesting our help. It is a win-to-win strategy. I think they are in a good understanding.
Thank you so much.
Dr. Celia, can I come in?
Yes, please.
I think Dr. Celia, I think NBPGR or IMR may take initiative to introduce this case 23-6 into our germplasm stock. Other than this, any other lines which are reportedly resistant to this particular disease, maybe I think we may put in our efforts to introduce and keep in our stocks. I think this point may kindly be noted.
Yeah, good idea.
Good idea. Yeah. We will tell the germplasm exchange and policy unit. Yes.
Yeah,
yes.
we must introduce.
Yeah. This is a very good point. When I was in IIT in Nigeria in 2010, then I went to Nigeria to Senegal, and there is a hopping flight where everybody from Benin also got down. So how the germplasm are there phytosanitary certificates are mandatory to move from one country to another country within the African continent?
Yes, that is very true. Between any country is within the region, we do first request import permit. We carefully look into whether this particular disease is there or not. Most of the Southern Africa country, they do request the seed should be completely free from MLN causing viruses. So not just we just give the seeds, but we also test them before we handing over to our local phytosanitary agencies. So with that dual testing, and then they will also take the test in their quarantine facilities. Not just it is not that we are doing everything is wrong, but they also take precautions in their countries. They do sincerely. Yeah.
Thank you, Dr. Suresh. Is there anybody wants to ask any clarification or any question? In the chat also, there are no questions.
Yeah, if any question, any information is required, I'm more than happy to answer by email or phone. Yeah, whatever the information I can share, it is quite helpful to the people.
Dr. Suresh, I request you to kindly share the write-up, and we will publish it in as a special issue for the international conference. We will all these virtual talks. We are actually going to make a compilation, and I think mostly we will include in a special issue. Kindly share the write-up at the earliest, if possible, in the journals format, please, Dr. Suresh.
Yeah. This, of course, this information has been published already in virus research, but however, I can. I will try to share is most possible. Yeah.
No, please do that, sir. That is, because you have covered a very good information. It's a significant disease for India. So I think it will be useful if you share your write-up. It is required actually to publish. Thank you, Dr.
Yes. Thank you for your nice support, encouragement, and to have this session with all these eminent audience. And I do sincerely thank for the organizers for giving me opportunity. Especially Cecilia Madam, as you always want to be having. Yeah.
I have a special thanks to Dr. Celia Chalam and also Dr. Suresh. Now, I think if there are no further questions, we move on to, formal vote of thanks. We have Dr. Suresh from Dr. Ramesh from Coimbatore, our Plant Protection Association of India's executive committee member. I think he'll, formal vote of thanks is given by Dr. Ramesh. Ramesh, please.
Good afternoon, everyone. It's really a great pleasure having Dr. L.M. Suresh, lead Maize Pathology, head Emerald Screening Facility, CIMMYT Africa, for joining today's webinar. That is the 8th lecture on PPAI Golden Jubilee lecture series. So, sir, on behalf of the Plant Protection Association of India, I would like to extend my sincere gratitude to you for the very informative talk on biosecuring Sub-Saharan Africa from Transboundary Maize lethal necrosis disease. Actually, the issue of MLN disease and 1 decade long efforts in successful combating the disease is very wonderfully covered in the lecture, sir. We are thankful for your efforts in giving detailed account on the development of maize tolerant hybrids, screening them in hotspots, describing about MLN phenotyping facility, quarantine facility, disease diagnosis, surveillance, phytosanitary protocol, and all the wonderful information given during this lecture, sir. And again, the interesting information on approaches for integrating various stakeholders of maize industry to combat the disease is very well covered with the help of policymakers, seed production agencies, maize farmers, especially in a community level participation approach, how successfully the CIMMYT has eradicated our CIMMYT effort in combating the disease is very wonderfully given, sir. So the plant protection scientists, especially the quarantine scientists, are really greatly benefited by this wonderful lecture. Sir, thank you very much for sparing your valuable time and sharing your knowledge through this webinar. We are grateful to you, sir. And I extend my sincere gratitude to Dr. B. Sharad Babu, President, Plant Protection Association of India, for organizing this PPAI Golden Jubilee lecture series. Actually, talks from eminent scientists from different countries like US, Australia, Singapore, Africa, on cutting-edge technologies like CRISPR-Cas, space entomology, precision plant protection, and recent trends in biosecurity, all very well arranged and organized by Sharad Babu, sir. Sir, really, the plant protection scientists are grateful to you for your efforts in organizing this event, sir. And a special thanks to Dr. V. Celia Salam Madam, Vice President PPAI and Head Division of Plant Quarantine NBBGR in New Delhi, for moderating this lecture series and giving valuable inputs, and again helps in organizing this event. Thank you so much, ma'am. I sincerely thank Dr. R. Jagadishwar, Director of Research, PJT SAU, and Vice President PPAI for his great support in organizing all the webinars and similar events. I mentioned my deep sense of appreciation for the conveners, Dr. L. Saravanan, Dr. B. Parameshwari, Dr. Prasanna, and Dr. Bhaskar for their efforts in organizing the event. I also wish to express my sincere gratitude to all the Executive Council and office bearers of PPAI for facilitating these events. I must thank the organizing team, officiating staff of PPAI, and NBBGR Regional Station Hyderabad for their efforts. And I would like to especially convey my sincere thanks to Dr. Anita Kodaru, Principal Scientist and Head NBBGR Regional Station Hyderabad, for sparing all the facilities to conduct the event. I sincerely thank all the esteemed delegates of this today's webinar for their participation and contribution to make this webinar a great success.
a galaxy of researchers and scientists from ICR institutes like ICR, IARI, NBPGR, IAMR were there, and researchers from SAUs like BJT SAU, ANGRO, PAU, and US Dharwad were present during this seminar. And most notably, international organization officials from ICRISAT and even National Plant Protection Agency like NIPHM and other officials have joined this webinar. So thank you very much for all the delegates and participants for your participation and contribution. So thank you everyone once again for joining today's webinar and for making it a great success. Have a great day ahead. Thank you.
I once again thank all the participants. Thank you so much from the core of my heart from Plant Protection Association of India. I look forward to all your cooperation in the days to come, and we are having a wonderful. We are planning good lecture series. We have many significant people lined up for in the next few months. So let me not reveal at this point in time. I only request that please continue to participate in the series of lectures. Thank you very much, and thanks once again. Thank you all.
Thank you, sir.
Can you switch on their videos? We can take the screenshot.
Yeah. Can you all, whosoever is there, please switch on your videos. We will have a picture. Screenshot page. Thank you very much. Thank you all. We are now ending the.
Thank you.
Thank you, Dr.
Suresh. Thank you, everyone.
Thank you.
Thank you, Dr. Bhaskar, Dr. Celia, all my colleagues. Thank you. And yes. What this needs to be YouTube workload. Actually, I can paste it on the. Actually, desktop window is desktop just screen print screen so don't worry, it is all done.